Okay. Screen. All right. So we were talking about triacylglycerides, right? Okay. Ready for this? Everybody got their thing turned in or emailed, right? Some people are having an issue printing. So as long as you send it in to me and then put it in on my door, it's all good. Okay. So why did the melon jump into the lake? It wanted to be a watermelon. There was another one today. Where'd it go? Oh, what do you call a mischievous egg? Wait. Wait, no, it was terrible. It's a jokester? No, a yokester. That's what it is. It's a yokester. I can remember the thing. Oh yeah, that would be great to do iClicker. I had to have IT come out and fix iClicker in this room, but it should be fixed. That is a great question. No, you did not have to, because your test proved your presence on the day of the test. That's that's all right. Okay. Yay. Okay, it's working. See if you can click in. Yay. All right. Okay, so now we can really start. Okay, so the last thing we were talking about was chylomicrons, right? So when you think about fats in the body, I want you to think about basically 3 different mechanisms that have to happen, right? Number 1 is we can eat fats, right? And then the fats have to be brought into different parts of the body. We can make fats. When do we make fats? When do we make fats? High energy charge of the cell, right? I've carb loaded on pasta or whatever it is, right? I have a high energy charge. I don't need to make any more energy, but I have more glucose floating around. And so my body is going to say winter is coming and I need to prepare, right? And so you have to store that energy, right? Y'all like that reference, huh? Winter is coming. So that's 2. Okay, now what's the 3rd mechanism that we have to worry about? It's not intake or production, it is the opposite, which is degradation, right? Once we store it, then we have to say, okay, let's say we're under starvation mode, right? We're going on a low carb diet. What's going to happen? Well, the cells still need energy, so they're still going to have to metabolize the fats that are located in the body and get them out to the peripheral tissues, right? So that's 3 major mechanisms. Y'all with me? So the first one was, I'll repeat. First one was that we eat high fat foods or foods with fat, right? Then the fat has to get into the body somehow. The second one is that we carb load, right? And we're going to make fats. And the third one is that we have stored fats and we need to metabolize them. So we got all 3? Yeah? Okay. So the very first one, this is, we'll call it number 1. This is fats we eat. So fats we eat are eventually going to be transported through a chylomicron, which is what we talked about last time, right? That they're very large. You know, you've heard about high density, low density fats, right? The good cholesterol, the bad cholesterol, right? All that kind of stuff. So these are very high density. These are the largest kinds of vesicles that will transport fats in the body. Very large. So you got to think about what happens in order to make these high density. Oh, nope, that's not the one I want. This does it differently. Okay, so we're going to skip ahead a little bit and then we're going to come back. They organize this one differently. So if you eat fats, right, what does your body do in order to metabolize those fats? Anybody know of a relative who lost their gallbladder? Okay, so what are the dietary recommendations if you no longer have a gallbladder? Low-fat diet, a low-fat diet. So a low-fat diet, you and me, Sam, we don't, I guess we both don't have our gallbladders. So if you don't have a gallbladder, what does a gallbladder hold? Bile, excess bile. Is it the only place you get bile? No. What makes bile? Liver. Liver makes bile. So the liver makes bile kind of all the time. And all the excess bile that's not used goes to the gallbladder. So when you eat a high-fat meal, your body senses how much fat is in that meal. And then if it's high fat, your gallbladder contracts, sends bile to the digestive tract to solubilize, emulsify the fats, right? So if you don't have a gallbladder, you cannot make a large volume of bile enter the digestive tract. Your liver still makes the same amount of bile, but it kind of like secretes it all the time instead of building it up and waiting for a high fat meal, right? And that's why people who have gallbladder attacks have the attacks after they eat a high fat meal. So the problem with gallbladder is that the concentration of the bile can increase to the point where you have crystal formation. So then when the gallbladder contracts after a high fat diet, what happens? It contracts around not liquid anymore, but crystals, solid crystals. And depending on what, exactly nucleated the crystal tells you what's in it. But how painful is that? Like really, really, really painful. Like when I had my gallbladder attacks, I legitimately thought, I might die today. Like it was that bad. It was that bad. I was like, something is seriously wrong. This is not right. I need to figure out what's going on. So you have to think about basically what the body needs at that point. Okay. So we've eaten a high fat diet. We secrete lots of bile. That bile emulsifies our fats. So now the fats are no longer, they're kind of like free triacylglycerides, right? So a triacylglyceride is that glycerol molecule that's represented by the red, right? And then the fatty acid tails are the little squiggles. So it's a triacylglyceride. So how many fatty acid tails? 3. Now, you can't send this across the membrane as a triacylglyceride. It won't work. What do you need in order to get across the membrane? Free fatty acids or a chylomicron, but we're not at that point yet, right? We're just in the digestive tract. This is the epithelial cells in the intestine. So what's going to have to happen to these triacylglycerides in order to get them to pass? I need free fatty acids. So your pancreas is going to secrete lipases. These lipases are going to cleave the fatty acid tails from the glycerol molecules. Then the individual components can be shipped into the epithelial cells, then reassembled into triacylglycerides. Then those triacylglycerides are packaged into the chylomicrons. And remember, what do we find in a chylomicron? Not just triacylglycerides. What else do we find? What else is transported in a chylomicron? cholesterol, right? Triacylglycerides and cholesterol esters. Okay. Yeah. So where were we? I'm going to like jump back and forth, but it's okay. So now we're going to assemble a choliomicron. Choliomicron has a triacylglycerides on the inside. It has cholesterol. What else does it have? It's surrounded by a layer, not a bilayer, but a monolayer of phospholipids. Embedded in those phospholipids are lipoproteins. Okay, so now we're going to export this choliomicron out. We're actually going to export this choliomicron to the lymphatic system. What is the lymphatic system? All your lymph nodes, your lymph vessels, right? Lots of your immune cells travel through the lymphatic system. So it's another transport system within the body. Then once that gets in the lymphatic system to the subclavian area, then we're going to convert it from lymphatic to circulatory, and then from circulatory, it's going to go basically everywhere. Okay. Good so far? All right, so now we're going to go back. Yep. Okay, so now we're going to go to number 2, right? So we said 1, we can eat it, right? We'll turn it into a choliomicron and we'll ship it out. Where does it go when we ship it out? Like I say, you know, we made a choliomicron, we put it in the circulatory system. Where does it go? Go back. Well, no, it goes to lymphatic, then it goes to circulatory, then it goes to peripheral tissues. So peripheral tissues are basically, what is peripheral tissues? Like everything, like any cell that needs it, right? But then if you have excess, where's the excess going? To adipocytes, right? If you have too much of a high fat diet, your body's not going to waste the fat. Your body's going to store the fat because guess what? It thinks winter is coming, right? So it has to prepare. So it's going to store that excess fat in adipocytes. Okay. All right. So now let's go back. All right. So now what happens if you're not eating fats? Let's say you're eating carbs and your energy charge of the cell gets really high. What do you inhibit? What else? Krebs cycle. So you're actually going to make acetyl-CoA, right? You are actually going to start breaking down that sugar. Where you're actually going to see the bulk of the inhibition is in the citrate cycle. So if you inhibit the citrate cycle, we're no longer going to go through electron transport chain to make energy, but we're going to have acetyl-CoA. So what are we going to do with that acetyl-CoA? What do we say in Biochem 1? What happens to it? It just gets shuttled off to make fat, right? Triacylglycerides. So this is that pathway, right? So we ingest lots of carbs, turn it into glucose. Glucose gets converted to pyruvate and acetyl-CoA, right? Now, on the other hand, even if you ate a high protein diet, if you have excess proteins, those proteins get broken down into amino acids, which can then get broken down into pyruvate and acetyl-CoA. So this is going to happen if you have an excess of any energy source, if that makes sense, right? So we're going to make pyruvate and acetyl-CoA. The pyruvate and acetyl-CoA are going to be metabolized to make palmitic acid. So palmitic acid is an example of a fatty acid, right? So it's one of these individual fatty acids, right? Now, you're going to synthesize a bunch of them and you're going to convert them and make them into triacyl glycerols, right? So we have a glycerol molecule and we'll have 3 of those palmitic acids and we'll turn that into a triacyl glycerol. So now we're going to package these triacylglycerides along with some phospholipids and lipoproteins, and we are going to make something similar to a choleomicron, but not the same. We're going to make something that is smaller. So we call it a very low density lipoprotein, lipovesicle, however you want to think about that. But this is going to be much smaller than the choleomicrons. So you can actually tell what source the fats are coming from just based on how they're being transported. So the assembly actually occurs in a combination of the ER and the Golgi, right? They call that the endomembrane system. It's both the Golgi and the ER. And then put it in a secretory vesicle, merge that vesicle with the plasma membrane, and we release our very low density protein here. Okay, so now this can actually go and be transported to peripheral tissues, and this actually goes through the circulatory system. Okay, so depending on where your fat source comes from will tell you what kind of transport mechanism is using it. If your body makes it, right? If you're making it from excess carbs, excess proteins, it's going to be in something that's low density. If you are eating it, then it's going to be in something very high density, the choleomicrons, right? Make sense? How y'all doing this morning? I would kill for a cup of coffee this morning with like an extra shot of espresso. Yes, right? Right? I know y'all are with me. All right. That's a different chemistry of the body, right? Okay, so now, regardless of where you get the fats from, if you have excess, your body's going to store it, right? Winter is coming, let's prepare. So where do the excess triacylglycerides go? They are going to go to your adipocytes. Now, adipocytes are kind of cool in that if you look at the cell, right, this is one cell. The lipid droplets, it can be one or it can be a few, but the lipid droplets grow in size depending on how much fat you consume. So you don't ever actually change the number of adipocytes in your body. You change the size of the adipocytes, right? That's why liposuction is different, right? What does liposuction do? It actually goes in and removes adipocytes. It does not change the size of the adipocytes. Changing your dietary intake will change the size of the adipocyte, but not the number of adipocytes. Yeah, kind of like skin pigmentation. Everybody has the same number of melanocytes, but different levels of melanin are made in the melanocytes. That makes sense. Okay, so our adipocytes, most of the contents of the cell are those lipid droplets. And so much so, if you look at, when we did the lab, do y'all remember the lab in 437, Biochem lab? We did the lipid lab, and I showed y'all a histology slide. No, you don't remember this. I'll have to pull it up maybe at the end of class if you want to see. But if you actually stain them, what you'll see is basically it looks like open cells, but the majority of the cell is actually just fat on the inside, and the nuclei are smashed against the sides of the cell, and you don't see as many of the organelles, right? Because the lipid droplet is taking up so much space inside of the cell. From whatever chapter that was 13. So you can look at the different adipocytes, and you could see that adipocytes size vary pretty widely. Like this is pretty huge compared to a smaller one over here. And it's really all about how much lipid is inside of the adipocyte, right? And that changes the amount of fat inside the body. Okay. But we talked about this already, right? So now this is our triacylglyceride metabolism. So this was when we ate it, we turned it into a choleomicron, right? And then we can transport it to our adipocytes. But what happens on the other hand, this is like number 3. This is our third one. But what happens when you go on a low carb, low fat diet, or you get stranded in a cave for, I don't know, a week? What happens? What happens to the energy charge in your cells? It drops. What happens to your blood sugar? It drops. Okay, so if your blood sugar drops, what hormone do you release? Glucagon. I'm going to tell you the other one, right, that happens at the same time is epinephrine. Epinephrine also works, either or. You're gonna go through a different receptor, right? You go through the beta androgenic, beta receptor. I hate this pen so much because like it just drags across the screen. It doesn't actually like release when it's supposed to. I'm so sorry. Beta androgenic receptor. Okay. So either way, the signaling mechanism is going to meet in the middle, right? Remember those signaling cascades, and some of them were the same from Biochem 1. Remember that? Okay. So if we have glucagon, let's kind of stick with glucagon, but it would work if, you know, like the bear came into the classroom, the same thing would happen. So we have glucagon. Glucagon is going to bind to what kind of a receptor? A GPCR, right? A G protein coupled receptor. So that G protein coupled receptor is going to activate what second messenger? Cyclic AMP, right? Cyclic AMP is going to go and activate what downstream protein kinase A, PKA. All right, what does PKA stand for? Protein kinase A. It's a kinase, right? So if you know the names, you know what's going to happen. What is protein kinase going to do? It's going to phosphorylate something. Okay, so if you look inside of these adipocytes and you look in these lipid droplets, on the outside of these lipid droplets is a protein called perilipin. Perilipin has a phosphorylation site. So if you look here up at the top, is it phosphorylated? No. So this is in the absence of glucagon signaling or epinephrine signaling, right? It is unphosphorylated. But when we activate our glucagon receptor and go through cyclic AMP and activate protein kinase A, it is going to go in and it is going to phosphorylate the perilipin. Now, if perilipin is phosphorylated, its whole job is to prevent these triacylglycerides from being metabolized when they shouldn't be. So it's like a protective layer that coats the outside of the lipid droplet. And so you want those lipids to be protected and sequestered and left alone unless you have glucagon signaling. So the glucagon signaling phosphorylates the perilipin. When that perilipin is phosphorylated, it induces a change in a regulatory protein called G58. When G58 is activated, it binds to a lipase, a particular kind of lipase, the adipose triglyceride lipase. Now, how many different lipases do you see? We have adipose triglyceride lipase, hormone sensitive lipase, and monoacylglycerol lipase. 3. How many fatty acids are linked in a triacylglyceride? 3, right? The reason why. So the adipose triglyceride lipase will only cleave one fatty acid off of the triacylglyceride. When that first one is cleaved, now what do you have? Not a triacylglyceride, but a diacylglyceride. All right. So at the same time that cyclic AMP activated PKA, and PKA worked on perilipin, the PKA will also phosphorylate the hormone-sensitive lipase. The hormone-sensitive lipase works specifically on diacylglycerol molecules. So now we can cleave our diacylglycerol. We're going to release another fatty acid, and we're going to end up with a monoacylglyceride. The monoacylglyceride interacts with monoacylglycerol lipase. why is it okay that we don't have a mechanism to activate monoacylglycerol lipase? It only works if you have one attached to it. Well, it has to go through two other regulatory mechanisms before it gets to that point. Correct. It's being regulated by the two before it. So now, how is it, but it's terrible to have free fatty acids, right? You can't have free fatty acids in the cell, in the bloodstream. You cannot have free fatty acids. So when I say you have free fatty acids, it's a little bit of a lie, because inside of the cytosol, you have a fatty acid binding protein. It's called fatty acid binding protein 4. And it has like, it's kind of like cup shaped, and the fatty acid fits inside the cup. And then it has kind of like a swinging door that keeps the fat in the inside. And it transports it to the membrane where the fatty acid gets released. And then in the bloodstream, you have a protein called albumin. And the fats will then be transferred to the albumin to be carried in the bloodstream to go to different tissues of the body. So remember all the BSA that we used in lab? What does BSA stand for? Bovine serum albumin. Well, we have it too. The reason we call it BSA is because it comes from cows, right? But we have it too. Human serum albumin.
So it's just,
yeah. So if you buy it, you have to go and search albumin, and then you can get it from different sources. Familiar? Yeah, absolutely. Okay, so we have two little sections left. What time is it? I don't know what time. So wait, halfway would be, I want to give you all half, 35. Okay, so like we're gonna go a few more minutes, so we'll do like half of this section today, and then the next time we'll do half, and then the final section. Okay, so we said we have different kinds of lipids for different reasons, right? So for metabolism, what kind of lipids do we talk about this whole last section? What kind of lipids did we talk about? Well, choleomicrons hold this type of lipid. Triacylglycerides are for metabolism, basically. Now, cell membranes are different, are composed of different types of lipids, right? We want to have a protective layer around our cells to keep the outside outside and the inside inside. And we want it to be hydrophobic so that our cells stay hydrated. So the plasma membrane is that lipid bilayer that gives us that protection. Now, and I'm going to show you this video. I forgot, I wanted to pull it up and I couldn't find it right before class started, but I'm going to show you a video. You've seen the fluid mosaic model. Have you seen a live video of it? I'm going to play one next time. Y'all remind me. I'm going to play one next time. So we kind of think about the plasma membrane as something being static because it's always drawn that way in a book. But in the plasma membrane, you have these, they call them lipid rafts. And you have a particular composition of lipids for the cell membrane in general. And then you have these localized areas that have different lipid composition and different membrane protein composition. And they kind of float around on the outside of the cell. They're not stationary. It's very, very dynamic, the membrane. And we don't necessarily think of it that way. It is strong enough to give the cell its shape, but it's flexible enough to accommodate all kinds of structural changes that have to happen in order for cell signaling to occur. So we have three major types of lipids that are inside of our membranes, right? Do we have triacylglycerides? No, that is not one of them, right? We have glycerophospholipids, right? So remember when we were doing lab, what kind of lipid were we purifying? phospholipids. We were purifying phospholipids. There are sphingolipids and there's cholesterol, right? We've kind of haven't really talked too much about cholesterol yet. But if you look, membranes are about 37% lipids, right? And of that 37%, most of them are our phospholipids. The majority of them are phospholipids. And then you have the sphingolipids and the cholesterol. So we're going to talk about the most abundant one first, glycerophospholipids. So a lot of times what you're going to see in glycerophospholipids is that these are going to be involved in signaling, in signal transduction and all that kind of stuff. So if it's a glycerophospholipid, it has a glycerol backbone, but it can't be a triacyl glyceride, right? Because you wouldn't have a place for the phosphate head to go. So in these phospholipids, you have two fatty acids, and on that third carbon of the glycerol, that third carbon contains a phosphate head, and then you can have different things attached to it, right? So do this, right? And you're gonna have your double bond and then, right? Oh, come on, pen. Doesn't want to work, but this is our triacyl glyceride, right? So if I want to convert this triacyl glyceride into a glycerol, I remove one of the fatty acid tails, and then what do I put on it instead? Right, and then one of these is a oxygen, double bond to an oxygen, and then I'm going to put an X there. Okay, the reason I put an X there is because I have a phosphate group here. This is what makes it a phospholipid. But you have different classes of phospholipids. Those different classes of phospholipids depend on what else is attached to the phosphate group. So you could have phosphatidylcholine, so choline could be here. Phosphatidylserine, phosphatidylethanolamine, phosphatidylinositol. Depends on what that X group is, tells you what kind of phospholipid. So you remember PS, PC, do you remember that? PI from the lab, all the abbreviations, that's what all that is, right? But we just didn't cover that in lecture yet before we had lab, which is really, really sad, right? Okay, so the core of our phospholipid is that glycerol. We have two fatty acids, and then attached to the third carbon is a phosphate head group. So whatever is attached to that phosphate head group, the X in our diagram before, tells us what we actually have naming wise, right? So phosphatidylserine, ethanolamine, choline, inositol, right? Tells you what kind of phospholipid it is and tells you what it's going to be used for. So the general class of sphingolipids, these are the ones that are derived from sphingosine, but you know, inside of all of your nerves and things like that, you have that myelin sheath on the outside of your axons. Have y'all seen that in, I don't know, physiology or something, right? So there's a particular lipid composition to those sheaths that go on the outside of those nerve cells. And so you have sphingomyelin, so you have the cerebrosides, which is a sphingoglycolipid. So if it's a glycolipid, instead of being a phospholipid, what are we gonna have? If it's a sphingoglycolipid versus a sphingophospholipid, we have a sugar. We have a sugar, right? So once you start to understand some of the vocabulary, you're going to understand just inherently what some of these lipids are made out of. Okay, I don't want to run over on my time, so I'm going to pause right there, and we're going to switch over. Oops. Awesome. If you have a case study to turn in, please turn it in. And do slideshow. Okay, let me move this. All right. Let me move out of the way. Okay. All right, y'all ready? Mine all right.
Hi. Hello. We will be presenting case study 2, glycogen attempt.
How do I make it go? Oh, there we go.
Yeah.
Okay.
And I
Okay.
think you could either roll or do the back and forth with the arrow.
Okay.
So the patient is a 15-year-old man, so he's a teenager. So he has an average height and weight. So there's nothing like, it's not like fat or anything. So we came in and got complaining of nausea, vomiting, difficult walking, lethargy, and a darkened urine. Well, when the doctors looked at the patient's history, they found that he had persistent muscle cramping episodes. He was very easily fatigued. He had no known disease or illness. Didn't take any daily medication, and his brother and sister also had difficulty with exercise as well. So, when the patient explained the provocation, the episode when this happened, he was playing at school, intense exercise. So when his muscle cramping and fatigue started to happen, so then later on he caught a second wind and caused him to stay active for hours, a couple hours. So after he was finished playing, he had extreme fatigue difficulty, and he had extreme, like a low-grade fever. Yeah, dumps. So probable causes of this muscle cramps: dehydration, wasn't getting enough water; electrolyte imbalance, probably not drinking enough Powerade, and muscle fatigue. So the significance of that second when the patient is playing is that it's a condition where when the anabolic of ATP is not enough, they have to go to alternative means of storage for the fats, the fatty acids, and the lactic acid. That's enough. And so that's what you experience. It's like a switch. It changes to that. Sorry if I'm not it's just the general, it's basically it's like, oh, it's not enough. So it's got to change that to out. So let's now you stay at hours. But when after it was done, it exhibited the problem, made it even worse. Because it didn't have anything else. And it led to even worse symptoms. And the probable, as I said, the probable cause is the deprivation of three alternative energy sources such as fats and lactate. So the causes for the darkened urine, as the patient reported, is probably possibly kidney or liver damage or disease. And he also had a urinary tract infection as well. Suffered from severe dehydration. Also had myoglobinuria, which is myoglobin in the blood, it just goes down the urinary tract. So which diagnosed a rhabdomyolysis.
Rhabdomyolysis.
It was difficult saying that word. So when they've done the physical examination upon the patient, they found he developed, had normal developmental milestones. There's nothing wrong with development, so he was going normal with that. His blood pressure was about 104/58, so good. His average temperature was about 37 degrees Celsius, so no. And he had normal neurological function. He his lungs sound fine. He had normal organs. He had no out, like his liver. There's no swelling of the liver, so there's nothing wrong with the liver itself. Like he had no muscle fasciculations and no nuchal rigidity, which in that case is the stiffness of the neck. So the only abnormalities that they found in the patient was a mild tenderness of the muscle, the heart rate was 110 BPM, and the respiration is about 22 per minute. So yeah, harder than usual.
Yeah, so the nuchal rigidity would be like a, like you said, like a neck stiffness, which would rule out typically meningitis, which is a symptom of, like the nuchal rigidity would be a symptom of meningitis.
So the lack of, okay.
Yeah, sorry, the lack of, I didn't make that clear. The lack of nuchal, so meningitis usually comes with nuchal rigidity. The lack of enlarged liver rules out liver disease and certain types of glycogen storage disease. So there are many different types of glycogen storage diseases, and a lot of them do have symptoms of enlarged liver. So our patient did not have this symptom. Yeah, so hepatomegaly is the name of enlarged liver. The muscle fasciculations are the twitches or spasms in the muscles. So this is usually caused by dehydration, or it could be electrolyte imbalance or something like that.
Yeah, or electrolyte, or even, with a muscle problem, you don't know if it's a muscle problem or if it's a nervous problem, or if it's a problem in the nervous muscle communication. So looking at, do you have a neurological issue? Is it a good thing to evaluate?
So lab work analysis, the normal results were the complete blood count was normal. Serum potassium was normal at 5.2 milliequivalents per liter. Serum glucose, cholesterol, and triglycerides were normal. Blood urea nitrogen 45 milligrams per deciliter, which is a normal amount. Now on to the elevated result. The pigmented urine test, that's just anything found in the urine, like any kind of, I think I'll go into it later. The creatine kinase was 2,114 units per liter. Aldolase was 44.1 units per liter. Serum and urine myoglobin, and serum and myoglobin in urine, which is abnormal. Uric acid 8 milligrams per deciliter, which is abnormal, and creatinine is 2 milligrams per deciliter. And sinus tachycardia, which is high heart rate. Creatine kinase is an enzyme found in tissues that upon muscle damage. So anytime you find it's elevated in the blood, it's caused by muscle damage.
So what have we seen remember the last time? What have we seen?
Yeah, it's heart attack.
It's a heart attack.
So anytime you have heart damage, like muscle.
So you do have some similarities between skeletal muscle and cardiac muscle, right? And so if you have a rupture or a death of any of the muscle tissue, then what are you going to look for? Whatever the contents were in the cell are now in the bloodstream, which shouldn't be there.
Yeah, so aldolase is an enzyme that breaks down sugars in the muscles and liver tissue. So elevation would indicate muscle or liver damage. Another thing I wanted to mention was these creatine kinase, yes, muscle damage, but uric acid, creatinine, and serum and just the uric acid and creatinine. Those are both indicate those both indicate kidney dysfunction. So yeah, creatinine would be creatinine is produced by the liver, and anytime it's elevated is showing that the kidney is is having dysfunction. Urine casts are clumped protein wrapped cells, fats, and microbes. So yeah, this patient had casts, which is anything like any kind of solid particles down in the urine.
And think about why that might be really dangerous to have solid things inside of your urine.
Block the glomerulus.
Yeah, so a lot of the ductwork in the kidneys is super insanely small. So if you have anything that is a solid particle, you're going to start blocking those ducts, and then the filtration system's not going to work, and very, very quickly you can have a patient go downhill. Very quickly, right? Think about people who have to go on dialysis. How often do they go on dialysis? Every other day, right? So if you have kidney failure, you're in trouble.
So like maybe just a little bit of foreshadowing for our diagnosis. The casts are caused by the myoglobin in the myoglobin in the urine. That's what causes the kidney dysfunction. Yeah, so cast presence indicates kidney dysfunction.
So after analyzing his lab work, they diagnosed him with rhabdomyolysis, which is the rapid degradation of damaged or injured skeletal muscle. And this is usually provoked by overexertion. So whenever your ATP concentration is running low, or muscle injury. So when you have this complication, the contents of your muscle cells spill into the blood. So that can also increase your blood potassium, which can lead to heart rhythm irregularities, which I think is why he had the sinus tachycardia. It can also cause kidney damage. So like Matt was talking about, when myoglobin is leaked into the bloodstream, it goes to the kidneys where the casts are formed in the presence of acidic urine, and they obstruct the kidney tubules, like Dr. B had said, and that can cause renal failure. And that also causes dark colored urine, because myoglobin is a pigmented substance. So that's could be the reason for his dark colored urine. So to treat his, how do you pronounce it? Rhabdomyolysis. They gave him IV fluids, bicarbonate, and mannitol, and the patient's symptoms did subside, and his ion and enzymatic levels were also stabilized. So the IV fluids they administered helped to flush the myoglobin from his kidneys. The bicarbonate lowered the acidity of his urine, which would decrease the further formation of those casts, because myoglobin in the presence of acidic urine, that's whenever the casts do form.
So I have one question that they didn't really talk about this very well, I thought, in the case study. But why, when you have muscle cell rupture, do you see an increase in acidity? Anybody study that? So, yeah, and think about what's happening in those muscle cells too. What are those muscle cells releasing when they lyse? They are releasing cytokines. They're releasing tons of cytokines. They are not happy. They are doing this really. So yes, let's talk about acidity. But I do want to touch on that too. They didn't talk about that very much at all in this, but you do have a huge inflammatory response that's going to occur if you have cell lysis. Cells are dying. Huge response. But what acid is going to build up inside of the muscle cell? Lactic acid. So when your muscle cells rupture, what's going to happen to the acidity of your blood? The pH goes down, right? You have an increase in acidity, your pH goes down. So you have all kinds of crazy pH things happening in the urine, in the blood. I mean, like this. Thank goodness this kid's at the ER. Go ahead. I'm sorry.
So they also gave him mannitol, which is a diuretic, to help increase his urine frequency. So that would also kind of help speed up the flushing out of that myoglobin, so no more casts would form. They also wanted to keep doing more testing, which included an ischemic exercise evaluation, skeletal muscle biopsy, ultrastructural cell microscopy with the transmission electron microscopy, and myophosphorylase staining. So these results of the ischemic exercise test showed flat lactate and pyruvate curves, which is not normal, but it also showed a normal ammonia curve, which is normal for any strenuous exercise. The ultrastructural examination of the cell, so when they looked at it under the transmission microscope, it did show a lot of intracellular glycogen, meaning that there is storage of glycogen in those cells. But they stained for the myophosphorylase, which is the enzyme that breaks down that stored glycogen, and it didn't show any. So
why are you gonna have this flat lactate and pyruvate curve?
So no glycolysis, no pyruvate, no Krebs cycle, no lactate.
Yes, exactly, exactly.
Oh, sorry.
Can you explain the ischemic exercise? Why they do it?
Yeah, so an ischemic exercise test is the primary test that does that usually diagnose glycogen storage diseases. So what they do is they'll like do blood draws from your arm. So they get like the baseline for all of your levels, and then they'll put a blood pressure cuff on your arm and then make you like squeeze a stress ball for 2 to 3 minutes. And then after that, they'll take the blood pressure cuff off and then redraw your blood to see how your levels reacted to that exercise. So ammonia is indicative of significant exercise. So like if you were doing this test and it didn't show any ammonia, then you would have to do it again because that means you weren't working hard enough.
But if you weren't working, you should have low lactate and pyruvate.
Right.
But if you are working, your cells are working and they're not getting more oxygen, right? You're doing it in the absence of oxygen. Cut off the blood flow, right? Your lactate and pyruvate should go up.
So with him, with his results of his ischemic exercise test and the lack of the myophosphorylase enzyme, they diagnosed him with McArdle disease, which is glycogen storage disease type 5. The myophosphorylase is the muscle glycogen phosphorylase deficiency. Like the deficiency in the myophosphorylase is what causes the McArdle disease. And it's an autosomal recessive mutation of the muscle glycogen phosphorylase on chromosome 11, which is on the PYGM gene. So McArdle disease is usually pretty benign. The exercise-induced muscle cramping, stiffness, pain, fatigue, not anything super systemic within your internal. But symptoms do resolve with rest and exercise-induced almost immediate depletion of the ATP and creatine phosphate. So it super quickly depletes you of all of your energy sources, which causes you to switch your energy source mechanism to fatty acids or things like that. The biochemical logic behind it. So when we just learned about the glycogen phosphorylase removes and degrades glycogen from the chains, it produces the G1P or the glycogen 1 phosphate. So mature skeletal muscles have the glycogen phosphorylase isoenzyme, which is the myophosphorylase. And since that's deficient, there's no degradation of the stored glycogen in those muscle cells. So he can't get energy from his stored glycogen.
So then you see how important just having that 1 to 2% of glycogen inside the muscle cells is super important. Super important.
So they proved that he was storing the glycogen with the microscopy, so he can store it, he just can't degrade the
And
storage.
that's something we didn't talk about. Remember on that very last slide of lecture, I was like, look at all the disorders, right? And depending on which enzyme you have an issue with, will depend. Can you store it? Well, if you can store it, can you metabolize it, right? And so it was really important for them to say, does he have any stored glycogen in order to start to whittle down which disorder he actually had.
So since it's myophosphorylase defect, that's why his liver is not having any issues as opposed to like pretty much every other glycogen storage disease. Because I looked up, so there's like 12 other ones, and almost everyone has something to do with the liver.
Yeah, so he can store it, he just can't like excrete it out. So I think that would be why.
Ours is mainly muscle
But
focused.
yeah.
McArdle is really muscle focused.
Yeah, you have different enzymes. That's why
Right,
they call it an
So
iso.
it's myophosphorylase is specific to the muscle.
Right, that's why they call it an iso enzyme, because you have enzymes that perform pretty much the same function, but they're encoded by different genes. So you can inherit a mutation in the muscle gene, don't inherit a mutation in the liver gene.
question.
Oh, sorry.
How is his quality of life affected after his diagnosis?
I mean, like I said, it's kind of a benign disease. So if he does have like bursts of exercise, he is going to feel fatigue or cramping or stiffness. But other than that, he's pretty good. Other than him having that rhabdomyolysis, which can do some pretty serious damage, it is life-threatening. But with the treatment that they gave him, his symptoms did subside, so they know how to counteract that from being like getting worse.
Probably can't do much of that.
Yeah, I mean, he's not an NFL star for sure, but like
He
he can-
can, like a bookworm on
yeah, he
paper
can chill.
anyway, so it seemed like it would be
Yes.
okay. Probably because he can't run around, especially everything at that age, that's really, you know, like.
The
there's lots
ladies.
of, Like sports, and
Yeah.
this, and that, and he can't, you know.
Well, it's not that he can't, he just has to warm up properly. So he starts.
Yeah, but if he's gonna play football, that's like a 3-hour game. Hope he's eaten a lot of Snickers before.
He can function
Yeah.
somewhat normally, like, he can go play
Yeah,
basketball with his friends, or, you know, whatever it is, right? I would not suggest a pro athlete career, but you can absolutely
Well, you
exercise.
can do some like light exercise or stuff to get the proper warm, like. Even if.
So kind of going into like his lifestyle, like, you know, if it's going to affect his quality of life, his symptom management, since this is like an autosomal recessive disease, there's no cure for this, it's genetic. But there are things he can do to improve his quality of life, like having a high protein diet, which will improve his ability to heal wounds and form new tissues faster. A fat-rich diet, which will help with his like second wind ability, so have more fatty acids stored for, you know, energy reserves and things like that. Fat-rich diet is kind of like not the best stuff because it could lead to like coronary artery disease and things like that. But if he knows he's going to be physically exerting himself, he can take like oral glucose beforehand, or just have like a really good meal before.
So did anybody look up like the current diet recommendations? This is actually kind of an older case. Now they're recommending is a high carb diet, exactly right, but they've changed the recommendations. Yes, you do have to have protein, but to keep a lower fat.
Strongly advised against the high protein diet in some of the articles.
So you need protein in your diet. It's not that you shouldn't have protein, but a high protein is not necessarily the ticket, that it's really more of a high carb diet, so that you have more of those sugars readily available. Yeah,
Diabetes is hard to do.
Pick your poison, right? All right, go ahead.
So another thing he needs to do is avoid exercise bursts. So that kind of goes into like the warm up phase. Like he needs to warm up his muscles for a longer period of time before he does do any kind of activity. So that way he can achieve that second wind, which, you know, allows his muscles to go into like the secondary energy metabolisms and things like that. All right. So they asked about supplementing with creatine and vitamin B6. So creatine is a good supplement because it increases your phosphocreatine stores, which helps promote the rapid turnover of ATP. And it also is known to like increase muscle performance in various diseases that affect muscle size and function. It also is known to be pro-inflammatory against cytokines. It stimulates cellular proliferation, and it upregulates genes that help with like cell health, like cell division, things like that. B6 is also used as a cofactor in the muscles, and it's associated with skeletal muscle performance in relation to carnosine levels. All right, so next one was about McCarthy's patients and not having elevated lactate because they lack the monophosphorylase, so they're not able to break down their glycogen stores. So the monophosphorylase initiates glycogen phosphorylase to cause glycogen breakdown. So if you don't have the initiation of glycogen phosphorylase, you're not going to break down your glycogen, so then you're not going to have any lactate formation, so then you're not going to go through the Cori cycle, produce glucose and all that good stuff. So insulin functions promote storage of blood glucose and glycogen store. So overtreatment of insulin is going to cause your body to overstore glycogen. And since you can't break it down, then you're going to have this liver that's just, I guess I would say fatty, would be a fatty liver, because you're just going to have all this stored energy in your liver and no way to metabolize it. So So increased ammonia is a result of the breakdown of amino acids and the direct result of the deamination of ATP. So glycogen storage disease type O is technically not a glycogen storage disease, it's a glycogen synthesis disease because it inhibits your ability to synthesize glycogen, not break it down from its like storage, it's like the McAdams disease does. So a defective branching enzyme would cause you to have less non-reducing ends, so then it's going to have the same symptoms. You're not going to have as much readily available energy storage. Reduce your glycogen as efficient. Yes. Any questions?
Because we talked about just now the carbs diet, but if you eat too many carbs, then it'll convert to nitrogen and
I think it would be to like take those in so you can use them while they're still in the blood rather than after they're stored.
so like that would make a pre-workout rather than like all the time.
So it might be more indicated to eat high carbs if you know, like today I'm going to the gym. So like before you go work out, you want to eat high carbs. It would increase your blood sugar level, so that way you have that energy readily available for your muscles to expand. As opposed to like before you go to bed, eat a high carb diet, like a whole bunch of pastas. That way, because then you would just be storing all of that as opposed to using it for energy. That would probably be what they mean by a high carb diet.
And they, like they said in here, they also do recommend like moderate activity. So if you eat and then go like for a walk for 20 minutes, that could keep it in your blood long enough to use it then instead of storing it and using it later.
Yeah, right. So eat a high carb, but also take some moderate exercise to make sure you turn it.
So anybody know why they were talking about not a high protein diet?
Well, proteins get broken down that you don't want to have too much.
So if you break down lots of proteins, you're gonna release more ammonia. And so that can cause an issue. I think the number one thing is a lifestyle change in how you exercise, right? Because the number one thing you want to prevent in this disease, what are we trying to avoid?
Overexertion.
Overexertion, right? So if you go too far, you're gonna start damaging your muscles, which is not so bad because you're not going to die because your bicep isn't working properly. But what's the issue? What's the downstream issue? Kidney failure. Your kidneys fail. You got serious problems, right? So it's important for these patients to know where their limits are and to stop before they go too far, right? And so lifestyle change. Probably number one.
Do you think anything about, like, I noticed the temperature was kind of high, and then vomiting, like, it said he was vomiting, like,
Vomiting could be due to like overexertion and dehydration. So I mean, you're having like less circulation to your stomach and your intestines, and you've probably got a lot of inflammation in his muscles, and he's got like this huge cytokine release. He's got all these things. So I mean, I would imagine he would be a little nauseous. So the nausea probably ties into the overexertion, I would say. What was the first part of your question?
like high heart rate, and he also had like a temp, like a fever.
So the high heart rate, I think, is because of that rhabdomyolysis. So whenever the potassium gets into your bloodstream, it can cause heart irregularity. So I think that's what that's from. Also, I also converted 37 degrees Fahrenheit Celsius to Fahrenheit, and it was 104. But I looked up what the normal body temperature was in Celsius, and 37 is normal.
So you had it was. So why would you have a fever? So what causes the fever? It's your immune response. It's primarily your immune response.
So in the study, it said that a high fat diet can also be used, but was controversial.
Right.
And it had like some drawbacks. What would some of the drawbacks be for a patient with McArdle's?
Like for the high fat diet,
Yeah, for high fat
I mean, prolonged high fat consumption can definitely lead to like coronary artery disease. So like you don't want that to be your main lifestyle decision because it's gonna block up your arteries.
This condition affect the mental status of patient.
I did see that McArdle disease does lead to some sort of like depression and anxiety in most patients just because like they can't exercise, so they feel like they're restricted a lot of the time. I did notice that whenever I was researching it, but I didn't see anything about like some other neurological issues.
And then if you're anxious to exercise, I mean, if you're anxious about like getting sick from exercise, then it's gonna just cause like this cyclic cycle of anxiety. You know, you don't want to go out and do things, like you said, depression.
But is there like if you had this patient admitted to the hospital, would you expect this person to be like in a delirious state or in a,
Definitely.
Yeah, if they can't control their blood sugar, for sure.
if they can't control their blood sugar, but did you, when you were reading about the patient, right, they said that they were they were
Hard to
in their
wake up.
consciousness, that they were
Heart arousal.
heart arousal, but once they did, they were able to communicate. So there wasn't a whole lot of, like, if you're vomiting, if you're really sick, you're running a fever, yeah, you're gonna be a little bit out of it, but you're not gonna be completely incoherent, right?
I just wanted to take a break. That glucose, or
But where's the glucose coming from?
The liver.
Right? Right, that's other storage. Right, so it's important to kind of think about that, like, because this is a mutation in the myo version, the myo isoenzyme, you're not gonna have the neurological issues that you might have in other.
I just think the bottom one, the Anderson, whatever, it was like babies don't grow.
Oh, very, very serious.
Like she was saying, this one's like still serious, but more.
But the reason this one was picked is because it's statistically more likely, you know, so of all of them, well, like, it's still rare.
Yeah.
They're all relatively rare, but yeah. Okay, what other questions?
So they don't find that the resulting conditions from the defects with the branching enzyme were worse than what you would see with McArdle disease.
So the branching enzyme probably would be worse because it would be more of a systemic disease as opposed to isolated to the muscles.
I did see online, I couldn't find anything that stated it for humans, but I did see that bulls that have this condition are at risk for like decreased movement. They're more likely to die. They have symptoms of what would be similar to like muscular dystrophy, like very severe compared to what was the heart disease. But I didn't know if that would overlap with.
Should.
It seems scary. I'm scared. It's like hard.
Yeah.
It's like having, failure to grow. Especially
Your liver is so important. Your liver is so important. I think it's one of the most overlooked organs in terms of what it does for your body. Detox. Yeah. all that. So what about, what? No, it's about something completely different. So what about the family history? Right?
So the brother and sister.
He's got brothers and sisters. Tell me about the family medical background.
So the parents are regular, but since this is a autosomal recessive, it means both of the parents are heterozygous with the mutation. So probably their kids will have it.
The parents could also have the mutation, but they do live a very sedentary lifestyle. They have office jobs, or they don't move around a lot. The brother and sister may have the disease, but it comes in later. It comes in during adolescence.
Yep.
I didn't find any evidence on why it occurs in adolescence, but there's like a theory that like children have infatigable muscles. So like there's like different mechanisms in their muscles for like energy breakdown and storage and things like that, as opposed to when you get adolescence, you get to like
entirely possible that the other siblings have it, but have it just one copy, right? Or they could have two and it has manifested in. So one thing that you would advise the family at that point, right, is to do a genetic test for the other children so that you know ahead of time. Something you really need to start about with this. Well, I would say treating early would prevent major kidney issues.
But you
Typically,
can-
what you see, like,
But you would treat once the onset hit. So like, if
Yeah.
you know that there's a chance that I could have this and I went and ran a mile earlier and now I feel really bad, then
You're like, oh, you
treat
got
it early, treat it early to prevent.
Right, not let the kids stay at home in bed, you know, like, stomach ache all the time, I mean, you know, kids nauseous, I don't wanna go to school. How common is that, right? You know, so you might take that information differently as a parent if you knew the genetic history and you need to watch out for it. Be more careful first. Right, and then you can teach them, okay, you need to warm up, you need to, you know, yeah, absolutely, absolutely. You know, you really want to, because it's like that cumulative damage to the kidneys can be really, really life threatening later on in life, right? When kidneys maybe aren't functioning as well as they have in the past, so you have to worry about that.
Not warm up, you know, your limits and how not to push yourself too far. They can prevent a lot of future damage. And later on, in life, you'll be better off later on in life.
It might even help, but like the crash, if as a kid they learn how to do it, they might grow up and like, oh, this is what I have.
But it's just how it is.
Yeah.
Earlier in the passage, it said that patients didn't have any muscle manipulations, but why, since all the muscles are like so effective?
So they don't have twitching. So twitching is a different neuromuscular junction disorder. So you have multiple disorders that could lead to muscle fatigue, right? If your muscles are constantly twitching, right, they could run out of glycogen, they could run out of everything just because they're constantly twitching, right? So if you have somebody with like a seizure, you can have symptoms of rhabdomyolysis because those muscles are constantly working, they're constantly seizing, right?
I've experienced something like that.
So you have all that muscle contraction. And so it's important to know if the muscles are just over contracting due to a neuromuscular problem, or if they're contracting only when they're supposed to, but it's something inherent inside of them. So it gives you that diagnostic. Yes, absolutely.
So for the last question, not like the discussion questions, but your recommended warm-up exercise, and it looks like that was more aerobic activity, so low intensity and not like this spurts of like high activity. So after the warm-up occurs, does that mean that these patients can't and so technically like the anaerobic activity where you've got like the short bursts of intensity?
Right, so the warm up would use your readily available energy, and then once you finish your warm up, that would put you in the second wind where you're metabolizing another source of energy, so like a fatty acid metabolism, something else that would produce energy for your body other than your glucose source because you can't use them.
So yeah, so one of the recommendations is to do like light bodybuilding, light weightlifting. Because you can build up your muscle mass. Now I'm not talking extreme weightlifting, right? Right, exactly. But you're gonna build muscle strength, and at the same time, you're getting your muscles used to using your alternative sources, but always after a work. Absolutely. Right on. All good? All done? We finished my work, so we should be on off. these kind of things, if y'all a deeper understanding of how to link it to what actually happens to a person and what are you gonna tell them and how. Oh, and like, I don't know about y'all, but you go. If you have too much glycogen, if you have muscle spasms, I was like, well, I read it to my dad, and he was like, you're just going like, I think it's not that big, and what it actually was around in my life. So, you're right.
In my eyes sometimes. Twitch.
Yeah, that happens to a lot of people. Yes, it's always fun. Well, and I'm not tall, like, we have it. Yeah, we have it. I'm like, on it for hours, I'm like, oh, crap. I'm like 30 tabs open. But that's, well, that's, see, and what I like so then it's even worse, because it's more well,
Oh, I know. I don't have class tomorrow. I'm gonna yeah. And I was like, I have to write a letter.
Oh, baby.
Oh, yes, right.
Now you understand, right? All the challenges that instructors have to have. All right, yes. Yeah, if you just